Perkenalkan, kami dari kelompok 1, kami akan melakukan eksperimen pembuatan tempe dengan kacang kedelai dan kacang merah menggunakan Rhizopus Oligosporus untuk memenuhi tugas mikrobiologi. Izinkan kami memperkenalkan diri.
Ya, kenalin, nama saya Allestito M dengan NIM akhir 073.
Saya Aisyah Rizqiyah A dengan NIM akhir 075.
Nama saya Jeshica Maia P.S dengan NIM akhir 089.
Nama saya Mierelle Cornelia dengan NIM akhir 90.
Nama saya Jeremi Bidang dengan NIM akhir 100.
The goal of this experiment is, first, knowing the process of making tempeh using soybean and red bean as a raw material. Second, comparing the characteristic of tempeh produced from soybean and red beans. Last, comparing the characteristic of tempeh produced from fermentation using yeast and flour yeast. Tempeh is one of Indonesian traditional fermented product that has been widely recognized as a highly nutritious and affordable source of vegetable protein. Typically, tempeh is made from soybean, glycine max, fermented using Rhizopus Oligosporus. Tempeh is a traditional food of the Indonesian people since centuries ago, especially in Central Java and Yogyakarta. This traditional product is very beneficial for health because it contains essential nutrition like carbohydrate, protein, fat, vitamin, and mineral, and superior bioactive compound.
Tempeh is a traditional Indonesian food product made through the fermentation of legumes, most commonly soybeans, using mold from the Rhizopus genus, especially Rhizopus Oligosporus. This fermentation process enhances digestibility by breaking down complex compounds into simpler nutrients, while also enriching the food with protein, fiber, calcium, vitamin B, and iron. In addition to its nutritional value, tempeh exhibits functional properties such as antioxidant activity and natural antimicrobial effects. Rhizopus oligosporus plays a vital role as the fermentation starter. It produces proteolytic enzymes that break down soybean proteins into peptides, while contributing to the compact structure, unique aroma, and microbial safety of the final product. Soybean tempeh remains the most common type in Indonesia. It is characterized by a firm texture, uniform white mycelium, and a distinctive fermented aroma. Notably, it is also one of the few plant-based sources of vitamin B12, making it especially valuable in vegetarian diets. Meanwhile, red kidney beans are a promising alternative base material for tempeh fermentation. Rice flour can serve as additional carbohydrate source, potentially influencing mold growth, fermentation speed, and the sensory characteristic of the tempeh. Although less widely used, they are rich in iron, dietary fiber, and plant-based protein. Utilizing kidney beans supports local legume diversification and enhanced nutritional potential of tempeh. An important variable in tempeh fermentation is the addition of rice flour. The combination of different legumes with rice flour presents an opportunity to modify and optimize the texture, flavor, and nutritional content of the final product. Lastly, fermentation is influenced by the key factors such as temperature, humidity, fermentation duration, and the quality of starter culture. Optimal fermentation typically occurs over 24 to 36 hours at room temperature. Clean handling and active Rhizopus species are critical to achieving a safe and desirable end product. The ingredients you need are kidney beans, soybeans, water, Oligosporus yeast, and rice flour. And the tools are container and container cover, digital scale, boiling pot, portable stove, tray, strainer, plastic, spoon, and basin. First, measure 150 grams of soybeans and another 150 grams of red kidney beans. This will be our two main substrates. Next, Prepare 2 pots, one for soybeans and one for kidney beans. Pour in enough water to fully submerge each type of bean. Then, boil the soybeans until they reach a rolling boil and the red kidney beans for around 40 minutes. Once boiled, drain both beans and let them cool. After cooling, peel the skins off as thoroughly as possible until most or all of the skins are removed. Now, soak both beans separately in water for 12 hours or overnight. After soaking, wash and drain the kidney beans, then spread them out on a tray. Place the tray under the sun and let them dry completely. For soybeans, wash them again, then boil for another 30 minutes. After boiling, drain and place them in a basin lined with tissues. Then, sun dry the soybeans until they are fully dry as well. Once the beans are dry, it's time to add the yeast. Mix the kidney beans with 0.3 grams of yeast and do the same for the soybeans. Make sure the yeast is evenly distributed. Now, take a portion of the kidney beans and soybeans and mix each with 1/3 spoon of rice flour. This will help us compare the effect of rice flour on the fermentation process. Wrap each samples, both with and without rice flour, in a food-grade plastic. Poke small holes in the plastic to allow for air circulation. Label each one according to its content. Now place the wrapped samples in a warm, dry place and leave them to ferment for about 48 hours. Make sure the temperature stays around 30 to 35 degrees Celsius if possible. In this experiment, we fermented tempeh using 2 main ingredients, soybeans and red kidney beans, along with 2 types of starters, pure Rhizopus Oligosporus and a mix of yeast with rice flour. Each treatment used a ratio of 0.3 grams of yeast for every 150 grams of beans, which is equivalent to the standard 2 grams per 1,000 grams of substrate. So, what influenced the success or failure of fermentation? Let's break it down. First, let's talk about the type of substrate. The results shows that soybeans are much more responsive to the fermentation than the red kidney beans. With soybeans, the mycelium grew faster, more evenly, and created a compact, cohesive tempeh texture. In contrast, kidney beans showed slower and uneven mycelium growth, and in the case of pure yeast, this happened because kidney beans have harder, slicker seed coats and contain anti-nutritional compounds like tannins, phytic acid, which can inhibit fungal growth. The second variable type is the type of starter. The pure Rhizopus Oligosporus culture worked very well on the soybeans without any additional ingredients. But on kidney beans, the growth failed completely. The mold couldn't colonize fast enough and was overtaken by contaminants including pests. On the other hand, using a starter mixed with rice flour helped the kidney bean sample successfully form into tempeh. The rice flour acted as an extra carbohydrate source and improved surface conditions, allowing the mold to grow more stably. What about the yeast dosage? The ratio used in this experiment, 0.3 grams per 150 grams of beans, follows the standard of 2 grams to 1,000 grams. It was sufficient for soybeans as fermentation succeeded without any issues. But since kidney beans are more difficult substrate, additional treatment like rice flour became essential, not a higher yeast dose. To conclude, This experiment shows that soybeans are the best substrate for tempeh fermentation, especially with pure yeast. Kidney beans can be fermented, but they require additional support like mixing with rice flour. Pure yeast perform well on ideal substrates, while rice flour enhanced yeast helps with challenging ones. All variables are connected, so for successful tempeh fermentation, it's not just about the starter, but also about choosing the right beans, applying the right treatment, and ensuring a proper fermentation environment. Now, let's take a closer look at the quality comparison between each tempeh sample based on 5 key parameters: aroma, taste, texture, mycelium growth, and final conclusion. We'll compare how each combination of bean type and yeast treatment performed by the end of the fermentation. 1. First, the soybean with pure yeast. This treatment gave the most ideal result overall. The aroma was natural, just like the store-bought tempeh, but more bland. The taste was mild and familiar, and the texture was firm and compact. The beans bound together well, and the mycelium grew abundantly, forming a solid white cover. This means fermentation was highly successful, meeting the expected standards of good quality tempeh. Second, soybeans with yeast plus rice flour. Adding rice flour to the yeast made a slight difference. The aroma was still neutral. The taste became a bit more bland compared to the pure yeast version. The texture was less compact, slightly soft in parts, and the mycelium grew moderately, not as thick and slightly uneven. Overall, this combination was still successful, just not as optimal as using pure yeast. Third, kidney beans with pure yeast. This was the only treatment that resulted in a total failure. Because contamination appeared early, there was no aroma, no flavor, and no texture to evaluate, and no mycelium growth occurred. Fermentation failed due to combination, likely because the kidney beans are harder for Rhizopus to colonize without additional help. Here where it gets interesting. Despite the challenges of kidney beans, the tempeh had a sharp and slightly acidic aroma for the kidney beans with yeast plus rice flour, a tangy taste that wasn't rotten, a solid texture, strong enough to fry, and thin, patchy mycelium growth, slower than soybean, but presents. It means that tempeh successfully formed, though with lower quality compared to the soybean tempeh. This table shows that soybeans are optimal for fermentation, especially with pure Rhizopus yeast. Rice flour helps improve fermentation for tougher beans like kidney beans, and while not all results are perfect, its treatment gives us insight into how fermentation works under different conditions.
For the conclude, making good tempeh is highly dependent on cleanliness, stability of the fermentation environment, and control of technical factors such as moisture content, temperature, and yeast dosage. The addition of rice flour to yeast can accelerate fermentation and produce a harder and denser tempeh texture than using a yeast alone. While for advice in the cleaning process, make sure the beans are completely clean from the skin to facilitate the growth of mold, and make sure the condition of the beans when adding yeast is completely dry to prevent microbial growth, because it can affect the fermentation process and the final product.